So today we're going to talk all about germline variants, which are near and dear to my heart. We're going to do a little bit of review from yesterday, just so that you have all the background you need to go deep into the algorithms in some of the subsequent presentations. So we're going to be starting today from that aligned recalibrated BAM that we talked about yesterday. And so at the top, we have our overall pipeline where you start with a tube of spit, blood, so on and so forth, goes through the sequencer, and at the end, eventually, you're going to get your high-quality variants. And so you can kind of make a matrix of different tools for different scientific applications. And so today we're going to talk about germline, SNPs, and indels, but there's also a somatic component for most of these tools, which we'll talk a little bit about next week, right? Yes. So there are a lot of different kinds of variants. Today we're going to talk about germline variants, which are the variants that you're born with. The vast majority of them you inherited from one of your parents, but in every person there'll be a small number, around the order of like 30 variants that are totally new to you. And not that they're deleterious necessarily, but just something interesting. And that's different from somatic variants, which are variants that your tissues will pick up over time as the cells divide. Also not necessarily cancerous, but just things that are different as the tissues in your body grow. And so there are a variety of variants of different sizes. So we're going to talk about short variants, and those are point mutations, or in the germline sense, we call them SNPs typically, for single nucleotide polymorphism, where the polymorphism comes from the fact that in the population, from a population genetics standpoint, that particular site is polymorphic, meaning it has different variations in different people. The community is kind of moving towards the single nucleotide variant or multi-nucleotide variant because it's less judgmental about what the frequency is in the population and whether you've seen it before. So those are just little single base changes in the genome, and they're typically pretty easy to identify in a lot of cases just by looking at your IGV screenshot down the line. And then there are also indels, which is a portmanteau of insertion and deletion, and that's going to be small numbers of bases that are inserted or deleted with respect to that reference sequence that Megan talked about last session. And typically the cutoff between an insertion or deletion that we consider to be a short variant and a longer one that we consider to be a structural variant is around 50 bases, which is kind of arbitrary, but it depends on the types of evidence that the algorithms use to discover those variants. So for the longer things, we won't be able to just look at the BWA alignments anymore, because that's so big that it's going to span more than a read length. And so we'll be using different kinds of evidence to our structural variation calling on larger insertions and deletions. We have tools for germline, copy number variants, and structural variation, but we won't be talking about them today. SVs are in beta. There's no workspace, but if you're really curious, the documentation is pretty good. Play around with that. And then there's a tutorial for GCNV that should be coming out soon for exome germline copy number calling. So this is our overall workflow for germline, SNPs, and indels, where the left panel is going to be all those pre-processing steps we talked about yesterday, ending in your aligned recalibrated BAM. And then today we're going to talk about the second and third columns, where we call variants and then do the refinement and post-processing. So the star of the show is the Paplutype Color, which is going to do the actual variant calling. It has 4 different steps, which James is going to talk about in a lot more detail later, but just to give you kind of an overview, we restrict our analysis to just areas that are interesting because we want to be efficient, and a lot of the genome is the same as the reference, honestly. We do a local alignment by a graph-based assembly, which helps us in complex regions of the reference or repetitive regions, or variants that are just kind of a little unusual in their sequence. We're going to assess the likelihood of each read with respect to the variant haplotypes that we discovered using the full sequence of the read rather than just the pileup of bases. And then we're going to do our germline genotyping. And so this tool has a lot in common with MuTech2, which we'll talk about next week for somatic. But the genotyping model here is where the tools really diverge. In the germline case, the vast majority of studies we do joint analysis, joint calling, a lot of people call it, with perhaps one or more samples of interest, and often a larger cohort, because one genome by itself isn't really that useful. Even if you have one variant that looks particularly deleterious in one patient, it's hard to say what the biological effect of that is going to be within its own signaling pathway, because you can't really derive that from first principles. And so a lot of the time we figure out what things do in the genome based on comparisons between people that have a particular phenotype and people that don't. A lot of this is with respect to a reference panel of healthy individuals like GATK and NOMAD, because we want to know if this variant has been seen before in a lot of healthy people, then it's probably not contributing to my patient's disease. But if it's something that hasn't been seen before, or maybe it's only been seen in 1 or 2 people, then that's a little more suspicious, and we should dig into those. So using this big reference panel or the rest of the cohort to cross-reference the frequency of variants in the healthy population is very important. If you have a family study, you can find out information about de novo variants by comparing the offspring to the parents. And again, in the disease case, a de novo variant could also be a little suspicious. And it's often very helpful to have a cohort or reference panel that's the same ethnic background as the samples that you're interested in, because perhaps if you have a Middle Eastern sample, it has some variants that haven't been seen before in a lot of the typical panels. Are those really going to be associated with the phenotype that you're seeing in that patient, or is it possible they're just common Middle Eastern variants that weren't picked up in your panel of mostly Europeans? So back in the day, we used to just process all the samples together with Paplutype Color. And so it worked pretty well for a while. So we'd put all the BAMs together, and Paplutype Color would assemble one big graph using all of the reads. And the more samples we added, the slower it got, because the graph got bigger and bigger and bigger. And there came a point where we just really couldn't do that anymore. We couldn't throw more memory at it. We couldn't wait, because it just kind of ground to a halt. And it got the job done. But every time we would add more samples to the cohort, we had to start again from the BAMs. And so we waited for so long to get this giant graph to evaluate and likelihoods to calculate. And then we had to do it all over again for all of the data, which was really disappointing. And so once we started generating the larger cohorts, I think it was probably tens of thousands of exomes, we moved to this new paradigm with GVCFs in sort of a 2-step process, because when James Van Der Beek has to recall all of his samples, he's sad. So the new 2-step GVCF workflow scales linearly in runtime and cost, because you only do the expensive Paplutype Color variant calling part on each sample once, no matter how many samples you have in the call set. And so a lot of times in the course of a project, you can have multiple waves of sequencing. You can have different grants that fund different parts of the project. You can have different collaborators that send their samples in at different times. And it's fun to be able to look at your data rather than wait for all of it to come in. And so at any point, you can take the samples that you have, and you can run the GVCFs generated through genotype GVCFs and start to look at your results. And once you add new samples, you just run genotype GVCFs again. And you don't have to waste all that effort recalling your original samples again.
You still have to do that for joint calling, right?
The genotype GVCFs part, well, and the genomics DB import are the parts that you will have to do again. Yeah. We don't currently have a solution where you can add additional GVCFs to a combined GVCF data store, which is something that we're working on. The Hale folks have something that's very promising, although it's not entirely finalized. But that would really, really help us with the N+1 problem, because already, once you get to these large cohorts of tens or hundreds of thousands of samples, the import and the joint genotyping becomes expensive in and of itself. And so if we can save some of the effort for that process, that's pretty helpful too. Right, so starting with our recalibrated, aligned, analysis-ready reads, we're going to start by going through all those 4 Paplutype Color steps. And in the current best practices paradigm of GVCF output for Paplutype Color, each sample is going to get its own GVCF or genomic VCF. We call it a genomic VCF because it gives information about every position in the genome, whether there's a variant there or not. And so in this example, the blue lines are going to give just reference information. because we didn't see a variant there, but it's important when you look at the whole cohort to know how many of your samples were confidently referenced. Because it's one thing if you have one sample with one variant and nobody else has data, because you say, oh, okay, that's a weird thing. But if you have one sample with one variant and 10,000 samples that do not have that variant, this becomes a lot more interesting. And so being able to hold on to that confident reference information as we go through the pipeline is very important. And then once we've combined together all the samples that we're going to analyze in this round, then we do our joint genotyping. Those reference blocks get evaluated if there are variants, or they get dropped if there are no variants, and we get more of a traditional VCF featuring only the variant sites. But are we done at that point? No, we are not, because those are just the raw SNPs and indels. Haplotype Color's responsibility is to be as sensitive as possible to pick up any variants that could possibly be real. But we know that there are going to be some things in there that are artifacts. They may look like real variants, but they're a result of mapping errors, they're a result of sequencing errors, and so on and so forth. And so another tool in the next step of the pipeline is going to be responsible for filtering out the things that are likely false positives. So, yes?
So in the genomic VCF, you mentioned every reference position is annotated, or I'm not clear what it means. So even if there is no variant, you say there is no variant in a location, or you track all variants?
If there's no variant, then we say that there's no variant. We kind of have this catch-all non-reference allele. James is going to talk about this a little bit more in the haplotype color talk. It's not as inefficient as it sounds with 3.2 billion positions in the genome. We have a couple tricks that we can make that a little bit better. But yeah, the goal is to be able to describe all of the data in that sample, whether...
So what's wrong in saying that if there is a variant, we record it, otherwise there is no variant? How is that different from specifically stating there is no variant in some place where you didn't see a variant?
Because when you say there is no variant, it's not clear whether there's no variant because everything was referenced or there's no variant because there was no data. So like if you imagine a scenario where you're combining together different exome captures, maybe you have one sample which is the only sample that has a target at that position. And so when you look at all your other samples, they just didn't have coverage there. And so is that, what's the allele frequency for the cohort at that position? If you assume that anything that's not listed is just reference, then you're going to be assuming information that you didn't have coverage for in your other samples.
So then the purpose of this is to distinguish between like missing data and no variant?
Exactly.
Okay.
Yes.
So we actually have been bumping up against exactly this in my lab, where we have 30x whole genome sequencing samples that we wish not to have to joint call with our other thousands of samples, because that costs a lot of time and money. And we need to do it really quickly to get results back, and so we've been calling just the one. And so then we've sort of like, when there's no call at a particular point, we're sort of guessing that it's homozygous reference because it's 30x WGS. How? Okay.
I mean, the 30x coverage it's very consistent in the vast majority of regions, but when you get into regions of high GC content, the coverage does tend to drop.
So I think we're doing this because it's only a specific set of variants that we care about, and they are variants that we have successfully called in other 30x samples. Okay, still not.
It's a risk. I've seen some things, so personally I wouldn't do that, but
You can join code just a region. You don't have to join code. You can join code just the specific region of your interest. You don't have to join code.
Right, that costs $1,000 on the cloud. Okay.
we should talk more. That's too much.
I'm struggling a lot with that right now.
Okay. Yeah, I will. We can talk in the Slack channel. Good. Yeah. So I mean, we have had a lot of these scaling problems in the past with our data, and we've moved past most of them. Our current project, we are hoping to combine 144,000 genomes, which is a lot of data. We think the final output is going to be less than a petabyte, which is good, but still a lot of data. So once we get our raw variants out of the haplotype color, we are going to need to filter those. And so we use somewhere on the order of 8 different features, like strand bias and depth and position of the variant within the read. And so given that our variants are going to be clustered in this multi-dimensional space based on those features, if you try and project them into 2D to do your own hard filtering by drawing boxes around variants that you think are good, you're going to lose a lot of precision. And so this is an example of sort of the hard filter scenario where you have one annotation on the x-axis and one annotation on the y-axis. And then we have the results from our filtering model shown there. And so you can try and capture all of those red dots that our model has declared to be pass with just 2 straight lines. Even if you got fancy and you kind of made like a function of those 2 variants, there's a lot of intermixture there because we have this complex space that we're now flattening into a plane. And so there are times that you have to do hard filtering because you don't have available resources, you don't have enough training data. Banu's going to walk you through a hard filtering tutorial later today. But in the vast majority of cases, we found that being able to use a machine learning model that can take advantage of the high dimensionality of the data produces much, much better results. That being said, with such a model, you do still need to pick thresholds. Like here we've picked a threshold, like if this x variable is greater than 5, it's probably good. But in the multidimensional space, you still have to make a decision where here's the line and we have things that are good and things that are bad. We'll talk about that when we evaluate the call set. So if you have access to additional information outside your cohort, like population priors, if you're working with a species, the vast majority of people at the BroadE typically work with humans, there are a lot of reference panels that'll give you a good estimate of what the population allele frequencies are. And you can use that data to help refine your genotype calls. If you have a study that has some population or some family structure to it, you can use that information as well. And by going through some Bayesian math that Takuto is going to walk you through this afternoon, you can use this to improve the qualities of your genotype calls. And so this is a particular example from some clinical data that I worked with a long time ago, where from the original calls that came out of our standard best practices pipeline, there were 417 potential de novo variants in this offspring, where we expect in a whole genome to be somewhere in the ballpark of 30. So that's a lot more than we were expecting, by like a whole order of magnitude. But then we took the 1,000 genomes data as population information, and that got us down to 17 de novos. And I think we also added the pedigree information to look at things that were also seen in the parents, give them more confidence, give less confidence, things that weren't seen in the parents. And then if you take only the genotypes of high quality at that point, you're down to 8 de novo variants, which is a much more reasonable number and a number that's easy enough to investigate from a clinical perspective, looking up those variants in the literature and assessing their effects and seeing if the genes could be involved in your pathway. Typically, people also do functional annotation. We have a tool that is out of beta as of 4.1 called Funcatator, which is sort of the next generation of Oncotator that you may have heard of from the old cancer genome analysis product. And so it's going to annotate variants with their predicted biological consequences based on common transcripts from RefSeq and so on and so forth. As you'll see, this is kind of the typical pattern for a call set, where a lot of the variants don't even happen in the gene, but that's kind of what we expect, right? Because most of the genome is non-coding, so most of the variation is going to be non-coding as well. Functional annotation will also point out things that are potential loss of function, like a stop gain, stop loss, start loss. Although this is not necessarily a smoking gun, again, it helps to compare that to your reference population because each person has somewhere in the order of 3 loss of function variants. But most of us walking around are perfectly fine. So whatever biological mechanism is involved in that pathway is compensating in some way. There are also other annotations that you can apply with Funcatator, with another annotation tool that could be helpful, like expression proportion of transcripts in different tissues. And then the MacArthur lab has a very robust tool specifically for loss of function variation prediction. And so the final step, once you've given all this information, annotated things, filtered them out, is to evaluate whether your call set looks good or not. And so whenever it's possible, a good study design typically includes some truth samples. We use NA12878 from the 1,000 Genomes Project, which is very well characterized, and then this newer synthetic diploid sample in pretty much all of our call sets because we have trustworthy variant calls for those, and so we can assess the sensitivity and precision for those particular truth samples against what's known, which helps us kind of get a ballpark of how good we think our call set is. So typically there's going to be two distributions of variance, sort of the true positives and the false positives, and depending on where you set your thresholds for your filtering, either through hard filtering or by picking some target sensitivity with BQSR, you're going to lose some of your true positives and potentially pick up some false positives. And so these are all just different ways of evaluating whether you picked a reasonable threshold is going to provide you with some high accuracy. Later this afternoon, we'll talk about important metrics in the call set, like TITV ratio, insertion-deletion ratio, and things that you can still use if you don't have high-quality truth data, just things that seem to be biologically true, and some target metrics. Because, again, you do have to pick this threshold at some point and say, you know, everybody on this side of the line is good, everybody on this side is bad. Because otherwise, you just have this ROC curve, which is a receiver operating characteristic back from old signal processing days. And it's going to describe how you ranked your variants based on whatever scheme that you've done to assign them scores. But again, you still need to pick a point on that curve. And so a good curve is going to have a very sharp drop-off, like that skier there is ready to take on, because I guess we're taking on challenges, we're good skiers. Whereas the things with the shallower slopes are not good results. But then to apply a threshold, you need to pick a point on that curve where you say, like, I want to pick up as many true positives as possible before my false positive rate starts increasing. And certainly that's a lot easier if you include truth samples in your call set. So that's an overview of everything that we're going to be talking about today. From our realigned, recalibrated BAMs all the way to our variant call set quality. So the subsequent talks will go into more detail in each of those steps, but happy to answer any questions you might have now.